I think we're live. Hello, everyone. Welcome to the second episode of the Future of Foraging seminar series. Great. Okay. Sorry, now we are complete. So I'm welcoming the two speakers for the session. So this session is on foraging at the limit, cognitive capabilities of birds and bees. And for this, we will have two speakers, Susan Healy and Lars Chittka. And maybe Rory, do you want to? Are you fully online again?
Yes, I'm here. I'm here.
All right.
Okay. Yeah.
Would you introduce Susan?
Of course. So, hi everyone. Yeah, I'm Rory Beresford. I'm one of the co-organisers of the seminar, and yes, our first speaker today is going to be Professor Susan Healy, who during nearly 40 years of research has made a brilliant contribution to our understanding of foraging and cognition. And her work focuses mostly on studying the link between behavioral ecology and neural processes in mostly non-model bird species in naturalistic contexts. In 1984, Suzanne graduated with a degree in zoology and physiology from the University of Otago in her native New Zealand. And she then moved to the UK, spending 8 years at the University of Oxford, where she did her PhD under Professor John Krebs. And this led to her first publication in 1989 on hippocampal specialization of food storing birds. In Oxford, she also began a long and fruitful collaboration with Professor Andrew Healy on spatial cognition in rufous hummingbirds, and then afterwards started as a lecturer at the University of Newcastle, beginning another long-term collaboration, this time with Professor Candy Rowe. Here she split her time between the lab, studying how hippocampal structure correlates spatial cognition with abilities of food storing birds, and in the field studying rufous hummingbird spatial abilities in the Canadian Rockies. She then continued her work on hummingbirds during 10 years at the University of Edinburgh and diverged from her normal study system of birds to also study sex differences in rats and humans. In 2009, she moved to the University of St Andrews, taking up her current position as a professor in the School of Biology, where she's continued to make important discoveries on learning and nest building and foraging decision making. And away from research, she's also published a number of books, the latest of which, Adaptation and the Brain, is out now.
Thank you. I will briefly introduce Lars, who will give the second talk. Lars Chittka is joining us from London, where he is a professor of sensory and behavioral ecology at the Queen Mary University. He was born in Germany, where he studied biology, and later obtained his PhD under the mentorship of Rudolf Menzel at the Freie University Berlin. And he and his lab study behavior in bees and bumblebees with a focus on ecology and cognition, looking, for example, at visual cues, especially the color cues that foraging bees use when visiting flowers. And his lab also has made some fundamental contributions to our understanding of animal cognition, for example, by demonstrating, he's like shaking his head. You cannot hear me. Can everybody else hear me? Okay. Okay, I'll go ahead. I'll continue and we'll figure out. So
you're fine. There's just a bad echo, so I think someone else has their mic on when they shouldn't.
Oh, okay. Yeah, I'm sorry. I'll just go ahead and please check your mics because I don't hear the echo. So yeah, he has made some fundamental contributions to our understanding of animal cognition by demonstrating the amazing learning abilities in bees. So for example, the use of tools such as string pulling and the ability to learn by observation. He has also received several awards and is an elected fellow of the Linnean Society Royal Entomological Society. This word is always hard for me, so, as well as the Royal Society of Biology. All right, I think we should start with our first talk, and we'll basically switch off most of the videos and see you guys later. And Susan, please go ahead and start sharing your screen. Oops. Now I'll mute myself.
Okay, can you see my first slide? Can I just get a...
Yes, I can see it. It looks good.
Okay, good. Okay, great. Thank you. Okay, thank you very much for the invitation to talk about foraging at the limit and hummingbird cognition today. Today, I want to think about these issues in the context of a behaviour that Darwin noted called traplining, which he saw as he walked around his sandwalk at Down House, that humblebees were apparently foraging in repeatable sequences at particular locations around his route. But before I get to that, what I want to do, first of all, is to acknowledge my long-term collaborator, Andy Healy, who was at the University of Lethbridge, and my partner in thought and experimental design. I also want to acknowledge the contribution of our species that we work on. It's the Rufous hummingbird, which is a sexually dimorphic hummingbird, and the female is on the left and the male on the right. And we do all of our work on the males. Okay, so we work in the West Castle River Valley in the Rocky Mountains in southwestern Alberta in Canada. The adult birds migrate from northern Mexico up to our site, which is in the middle photo here. And along the valley, for the 6 weeks that we are working on these animals, we put up these hummingbird feeders seen in the bottom right-hand photo, which contain sugar water, and a male will defend these very vigorously from everybody, including females of the same species. Okay, we can mark them, we can catch them easily and mark them with distinguishing non-toxic ink so that we know that we've got the correct bird when they work with us. And one of the best things about them altogether is that we can train them really readily. So, and by that I mean within an hour or two, we can get these birds feeding reliably from all manner of artificial, what we would call flowers. My pollinator colleagues would dispute that, but nonetheless, there are some examples shown here. And this is just to remind me to tell you too, that these birds are wild, they're free living. In the evening, when we've finished asking them our questions, we go back to the cabin for dinner, and they just carry on with their lives. And in the morning, we get up and go out and try again. So if they don't want to play with us, then they can forage from the flowers around the insects that they also take. Okay, but before I get to traplining, what I wanted to do, because I'm assuming that at least some, if not most of the people in the room, won't be massively familiar with the talents, the cognitive talents of these birds. So what I wanted to do was to just give you 3 brief examples of the kinds of questions we've been asking these birds across the time that we've been working with them. And I wanted to start with a question that many people think about when they think about hummingbirds and what information they might learn, pay attention to, and that's colour. And this wasn't quite our first experiment, but it was our second experiment. So some time ago, we wanted to know if we put out our patch of flowers, we've got 4 flowers here, only one of them has got food in. So that's designated by the pile of sugar there. And there's a pile of sugar water in that flower at the bottom left. So much so that the bird will not empty the flower when he comes in to feed from it. So he flies in, he has to figure out which flower has got the food, he drinks, and he leaves. And when he leaves, we switch that flower with another flower and we empty all of them. So the bird returns and none of these flowers have food in them. But the question is, which flower do they fly back to? And we just look at their first visit, and the data that we collected from that experiment is shown here. Along the bottom, the data are the first choice. So the bird comes in, visits one flower. It may, in fact, because it's empty, then go to the other flowers, but we're just interested in the first choice he makes. Up the y-axis is the percentage of visits the bird makes. And what we see here is in the pink bars, so the bars on the left, are when the birds go back to the flower in the correct location. So for the schematic I just showed you, it would be the bottom left flower. The yellow bar in the middle is the number of times that a male went back to a flower that was of the correct color. So that would be the flower that had been moved to the top right. And the third bar are when visits that the bird makes to the other two flowers. And what we found was that overwhelmingly, the birds went to the flowers in the correct, what we're going to call the correct location, didn't go to the flower with the correct color. So we then have spent quite a lot of time, as Rory said, looking at what these animals do with spatial information. And so here's an example of an experiment that we asked them this question. And here what we have is an array of 5 flowers. And the middle flower is the flower with the food in. The other 4 flowers have water in, and these birds don't like water, at least at our site, they'll spit it out. And what the bird has to do is to come in multiple times and learn eventually that it's just the middle flower that contains food. The ruler is there to tell you that on some trials, the flowers are very close together. They may be only 5 centimeters apart. And on other trials, the flowers are much further apart, and you'll see the distances in a minute when I show you the data. But once the bird has learnt that it's the middle flower that is the food that one that contains the food, we then move the flowers like this. So now there is still a 5-flowered array, but the flower that had been in the middle is now to the right. And there is a flower occupying the same, what we call location and space, that was occupied by that middle flower. None of the flowers are empty. We simply look at the first visit the bird makes. Which flower does he come to when he flies into this patch? And what we see here are the data. So if you look first of all along the bottom at the x-axis, you can see the spacing for the different trials that we had, so anything from 5 centimeters between the flowers all the way up to 3 meters 20. And then up the y-axis, we have the number of times the bird chooses a particular flower. The data on the left show that he chose the center flower, and those filled squares, show when he was choosing the center flower, and he did that more than expected by chance, up until the flowers were more than 40 centimeters apart. Once the flowers were more than 40 centimeters apart, what we found that the birds did is shown in the right-hand figure, where we saw that the birds went to the flower that is in what we were calling the correct location. So on the schematic, it would be on the left-hand side of those 5 flowers. So what these data showed us was that the birds are remembering spatial locations and positions of flowers, and it depends on what other information is around, what other cues they're using in order to determine which should be the flower that contains the reward. Okay, and finally, we also wanted to ask questions about whether or not these birds could remember when they visited flowers, because it's great to remember, to be able to remember where a flower is, but having emptied a flower, even if that flower refills, there will be some time delay before the bird should return to that flower, because if it returns too soon, it's going to find the flower empty. So what we did was to ask whether or not birds could remember when they had visited flowers. And the schematic of this experiment is shown here. And indeed, our PhD student, John Henderson, is also shown in schematic. And this is what it looks like to do field work with us, sit in a deck chair, drinking a lovely drink and watching flowers all day, that he did the following to. So there were 8 flowers, each of a different color pattern. And the bird came in and visited the flowers. When the bird first came in, all of the flowers had food in them, but not very much. So he had to visit more than one flower in this array to get enough food on this particular visit. Some of those flowers, so he'd drink and then leave. Some of the flowers, 4 of those flowers, Jono would put the food back in 10 minutes after the birds had emptied them. The other 4 flowers, Jono would put the food back in 20 minutes after the bird had visited them. So the bird flew in and through the day and found food or didn't find food in different flowers. Jono had to get out 8 little stopwatches for each flower and record through the day when the bird visited and when he had to put food back in. So we had absolutely no idea whether the birds could do this. So Jono spent 11 days at the first bird, and then we decided that we'd better do the same For the following birds, he managed to collect data from 3 birds through that season. They're only there for 6 weeks. And what he found is shown here. So we have 2 data figures. The top one returns when the bird returns to the 10-minute flowers, and the lower graph shows when birds are returning to the 20-minute flowers. And this is just one bird's data, actually, but the data for all 3 birds look essentially the same. The number of visits up the y-axis. And what we found was that the birds are returned to the 10-minute flowers just before or just after 10 minutes, more or less. The 20-minute flowers, on the other hand, they returned to the 20-minute flowers. The peak of their returns was very close to 20 minutes. So these showed that these birds could not only learn that some of the flowers were being rewarded about every 10 minutes and the other flowers being rewarded every 20 minutes. They did this concurrently for 8 flowers, okay, in the wild. They're busy displaying to other males and to females throughout all of this time. But nonetheless, and we discovered they'd done this by the end of the first day. But we had lots of data with our 11 days per bird. But over the time, what we've learned is that actually our hummingbirds can learn all sorts of different things. We've learned that they can learn and remember about space, they can learn about time, they can learn colors, and more recently we can show that they also learn something about numerical attributes of the flowers they're visiting. But what I want to do now is return to a context in which they are foraging and potentially, rather than just ask about their cognitive abilities, how about putting those abilities back into their foraging? And as I say, I want to do that in the context of trap lining. And actually trap lining has been not just in bees, as seen by Darwin and others since, but it has also been talked about occurring in hummingbirds. And indeed, in most of those situations, though, hummingbirds are thought to be of one kind of forager or another. So some species, for example, the hermit hummingbirds, are thought to be trap liners. Our and these are my hummingbirds, our rufus, are thought to be territorial birds, and that the two are different. Although in the purple-throated caribs, it looked almost as if the males are territorial and the females are trap liners, so potentially even within a species. But nonetheless, the thought has always been that these two foraging modes are separate, and that an individual is one or the other, not both. Nonetheless, it seems like it could be a really efficient foraging strategy if birds are using sequences and therefore following around flowers in a particular temporal and spatial repeated sequence. So we did want to ask whether our hummingbirds, given they could learn all sorts of other things, our territorial birds, whether or not they could develop a trap line. So the first time we asked this question, we did it in the following way, and this was done by a PhD student in my lab, Maria Tejo Ramos. And she put out a board, and you can see by the scale here between the distance between the flowers, the board is pretty small. It's still pretty big to a hummingbird, but nonetheless, it's pretty small. And we simply put out 2 flowers on this board. Each flower had enough food in it that the bird had to could get enough from one visit, but had to visit both flowers to get enough. So we looked at what the birds did with these 2 flowers, and if the bird made 9 out of 10 visits that were the same on consecutive visits, so bird comes in, visits flower number 1, goes to flower number 2, flies away, comes in, goes to 1 and then to 2, and so on, 9 out of 10 times, or 60 times to the board. Either way, then we added another flower, and we put the flower number 3 in between flowers 1 and 2. Again, we had the same 2 criteria, either 9 out of 10 consecutive visits that were the same, or 60 visits to the board. We added a 4th flower and did the same thing, and then we added a 5th flower. And the question is whether or not the birds developed a trap line. And I'm going to tell you now, yes, they did. But the data that I'm showing you here, along the bottom, we have our different patches. So we have 2 flowers, 3 flowers, 4 flowers, and 5 flowers. And up the y-axis, we have the mean number of sequences. if we just look at the dashed lines, these are all of the possible sequences that could be flown when there are 2 flowers, 3 flowers, 4 flowers, and 5 flowers on the board. If we look at the blue squares, these are the sequences that the birds flew at least once around the flowers. And then we looked at what the birds did, if they did anything repeatedly, which they did do. And then they had a very, very few number of track lines that they flew around these flowers. And those are in the green squares. Okay, the next question we might ask from these data is whether or not they were indeed efficient. So I'm showing you 2 here examples where there are 5 flowers. And if a bird was to fly in either of these 2 routes around the 5 flowers, that would be the shortest way to get round all 5 flowers and take food from all of them. And the distance of that is 72 centimetres on the board that we had. Here's a less efficient route that the bird could fly, where now I'm showing you the route that if the bird learnt or flew a sequence that mimicked the order in which we put the flowers on the board, would fly from 1 to 2 to 3 to 4 to 5. But you can see that the bird is crossing across. And this particular sequence is 20 centimetres longer than the one I just showed you. Okay, they didn't do that. What we found is that the birds flew the shorter sequences. Along the bottom, we have our 3 flowers, 4 flowers, and 5 flowers. Up the y-axis, we have the mean percentage of visits. And the different bars, the white bars, show when the birds were flying, how many, the percentage of visits where they flew the shorter sequences, and the dark grey bars are when they flew the longer distances. And at each of these different numbers of flowers, they flew significantly, they're much more likely to fly a shorter sequence than to fly a longer sequence. Okay, but as actually our reviewers pointed out, the scale over which we asked the birds that question was a bit, couldn't be further from reality in terms of the scale over which these birds forage. Nonetheless, I have to point out that even though the scale was really small, even there, the birds minimized the distance that they flew around those flowers. So for these birds, they really are paying attention. It's still costly to fly an extra 20 centimetres. But nonetheless, we did want to see whether or not we could ask this question and see what kind of answer if it was to be presented to the birds at a larger scale. And this we did with an undergraduate student, Mabel Barclay. So here the schematic is, we have 5 flowers out. Now there are 2 meters between each flower. And in this instance, we've put out 25% sucrose in each of these flowers, 25 microliters. And the aim of the game here is to try and get the bird to fly all 5 flowers. So that's how much we put out. We're trying to gauge how much he will take in one visit. 45 bouts. Every time there's a bout, the bird comes in and he can visit just 1 flower or all 5 flowers. There's always food when he comes back. And then we empty and refill each of the flowers between each bout. So the question was, did they develop a trap line? And what we see here's a schematic of what we found the birds did. So our the yellow circles are represent the flowers, and the blue lines represent the routes the most common route that the birds flew around these flowers. The size of the circle represents the flower that they're most likely to visit first. So the most common start flower and the flight paths around those flowers. The birds flew a trap line, and as I say, it was the most common one is the one that's shown here. The birds started at 1, flew to 2, flew to 3, flew to 4, and to 5. Which flower was one differed for each bird. But then we wanted to know is what they do with this information. Do they update it if things change? What happens if we modify what's going on? So now we have our flight. We know what the birds do when the birds have their 5 flowers. They've flown them for 45 visits. We know what their common trap lines are around those flowers. So we then emptied out the contents of flower 1 and replaced it with 5% sucrose. A little bit of sugar, but they don't like it if there's 25% in the others. And then we just let the birds visit the array and look to see what happened. What we found is shown here. So on the left-hand side in a paler yellow is what the birds, a schematic of what the birds were doing in phase 1. And in phase 2, what we found is that the birds very typically moved their trap line to start from flower 2. So the trap line, whichever trap line they'd been flying before, they started flying from flower number 2 and then flew in the same direction around the flowers. We had a bit more fun with this. So on some instances, the birds had flown 45 bouts. As I said before, when all the flowers have 25% in. And this time what we did, though, was we knew which flower they visited most commonly second in their trap line. And this time we put the 5% sucrose into the second flower in the trap line. What did they do here? Again, on the left-hand side, we have what the birds flew in their first phase matched up to what they did in the second phase when we had decreased the sucrose concentration in the second flower. This time, what we found they did is they redeveloped a trap line, but this time they reversed the direction that they were flying around the flowers, and they started from the same flower. So they started from flower number 1 still, but this time they then were much more likely to fly to flower 5 and go the other way around the array. So this here is, if I was going to talk about cognitive maps, and at least some of my group don't like me talking about cognitive maps, but here I might suggest that that the fact that the bird is able to be this flexible with its flight path might suggest that the animal has some idea of how the flowers are arrayed in space, and which is then the shortest route to go around these flowers, if number 2 has diminished in its reward. Okay, and finally, just to kind of go the other way, what happens when rewards get better in a trap line? And this time we looked at what happens if the birds had a trap line that we now identified which was the flower they're most likely to visit third, and we increase the concentration of the sucrose in that flower. And we increased it to 45% from 25%. And we looked to see what they did. And what they did was not as dramatic, actually, as it was when we really reduced the concentration of the flower. What they did was shown here. The first thing that we saw is that they did increase their starting at that third flower. So they switched a bit from starting at their typical flower number 1, but not significantly so. Okay, so they modified a bit, but not in the dramatic way that we saw, as I say, when we reduced the concentration. But what we also did see, though, is that and this wasn't a significant effect, that by putting a much increased reward into the third flower, they increased the number of shorter flights that they flew around those flowers, and they much more commonly included flower number 3 in the flights that they flew. Okay, so what we've done more recently, and we're only just really starting to work with this, we're now interested in how they're doing this. So how is it that they managed to fly around these flowers and figure out the shortest route around them? One way they could do that is every time they're at a flower is simply to look ahead at the next flower and go, that's the nearest flower, I'll fly to that. And they could be making that decision at every single flower. That seems less and not such a good explanation for what they do when they reverse the direction of their track line. But nonetheless, it's one way that they could work out how to fly to the nearest flower. So what we wanted to do was to see whether or not we could manipulate what they're doing by using a non-symmetrical array. And so we had this array out, which was slightly entertaining, making sure that the distances were right. And we've put the feeder up here because what we think is that often the birds come into the arrays that we put out for them from approximately where their feeder is. So a perch on a tree that's either near to or somewhere where they can see the feeder at their site. We take them down the feeder. It's never up when we're doing these experiments. So we put out this array. These are the distances between the flowers, and what we had expected our bird to do, if a bird could fly the shortest way around it, was to start with this flower that is nearest to the feeder. So the shortest route to the first flower is that flower there, and then to fly down to the flower that's 1.12 meters away, up to the right, and so on around, and that they would never actually fly that 4-meter flight at the bottom from flower number 6 to flower number 1. That's the longest part of all of this array, and that we assumed that they would finish somewhere at 6. So this was our ideal, and of course the birds didn't do anything. Well, they did a bit like that, but they simply didn't start at the flower that we thought they would do. They started at this bottom left flower and flew the shortest route around the 6 flowers. So the most common trapline is the one for 11 out of 13 birds, is the one I'm showing you. So these are the routes this is the route that they flew most commonly, and it was consistent across 11 out of 13 birds. But then what we decided to do was to, again, manipulate the contents and see what they did. So we replaced the contents of flower number 3 with 5% and let them fly again. And what we saw here is that the most common trapline was simply to leave out that flower and to fly a route that looks remarkably like this route, although, and I'm not showing it here because I'm showing you a schematic of what the birds generally did. At least half of the birds reversed the flight. So they flew actually from the bottom right flower around the other way. So reverse the direction of the arrows. And that's what at least half of the birds did. But we also found that one of the birds' most common trapline was this one. So this bird now started from the top right flower and flew this way around the array. And as it happens, this is the shortest way to get around the remaining 5 flowers if the bird starts from flower number 5. So as I say, we've only just started to really work with this non-symmetric array. But what I want to say about it here is that this is the kind of problem. This is the traveling salesperson problem. How to get around multiple locations in the most efficient way, in the short, and that isn't always the shortest route. For our hummingbirds, it is the shortest route, at least the way we've asked the problem of them. If you were to ask that of a computer, and it slightly depends on how many locations you've got, it results in the schematic down the bottom here, where the computer finds it inordinately difficult to do this. Our hummingbirds, on the other hand, seem to find this pretty straightforward. 45 bouts is about half a day or less, and well within that time, they are flying the most efficient, the shortest routes around the flowers that we put out. So I think I want to end here with my question marks after foraging at the limit, and whether or not our hummingbirds really are at the limit, either in their terms of their cognitive abilities, or indeed we might come to this in their energetic expenditure. And with that, I'd like to thank you for listening, and very happy to take any questions. Okay, and I will stop.
Thank you very much.
Stop sharing my screen. How do I do that? Oh, like this. Sorry.
Uh-oh. No, sorry. I think Susan dropped off. I'll take a moment to get her back on screen. And we have 2 questions from the audience. Just a second.
Apologies for my incompetence.
Okay. No worries. No worries. I actually don't know how you would stop sharing the screen. So, okay, we now have 4 questions. I will see. I think some are very specific, at least to your talk. So I think it makes sense to ask them right now. So the first one is by Aditya, and the question is: This is a question relating to the experiments that show that territorial hummingbirds can learn trap lines. Why do the number of sequences flown by the bird exceed the number of possible sequences?
That's because they, when we had the possible sequences, we had complete sequences. So for 2 flowers, a sequence actually could just be 1 flower. So even then, the bird could come on and fly to flower 1 and go away. Or it could come on and fly to flower 2 and fly away. It could fly from 1 to 2 or 2 to 1. So now we have 4 possibles because we included all of the incompletes that could fit into that.
And then the next question that has a large number of votes is from Chris Rogers. Do you think the birds are minimizing physical effort in flying or mental computational effort in planning the route? I wonder if there's a way to distinguish the two.
Yeah, if you have an idea, it's the kind of thing that we do. Yeah, I mean, we certainly have serious effort in designing the experiments to try and ask them the questions in the first place. And what we don't do is, which could be done in the lab that is not very easy in the field, is to really look at energy expenditure. But what we do see is they cut down the time they spend foraging, and it's one of the reasons they play our games, I think, is because even when we offer them sucrose concentrations that are less than what they might find in their flowers, we are very reliable. We turn up, we give them food, and we give them in quantities that are larger than most of the flowers. So during the time that we're there, we're very rarely likely to see them forage from natural flowers, even though the flowers are all around the ones that we're putting out. So they are definitely cutting down energy expenditure, even by playing our games. And cognitive expenditure, I know, that's a big question, isn't it?
I mean, we can return to this in the discussion as well, because it seems like, I think, a pretty good topic for the broader discussion as well. I'm just quickly looking if there's any, so it's a bit multitasking, so I'm trying to look if there are any other specific ones that should be answered right now. Here's one. Yeah, this one seems pretty specific. Do the hummingbirds notice when Henderson was refilling the flowers?
We don't think so, but it was a concern. But we've subsequently done that experiment again multiple times, where we mock fill every single flower, and it has no impact. So the birds are equally good at returning. The reason there's a dip, we think it's because the birds don't come in when he's there. But no, we don't think they were using him as a cue. But we also want to try that experiment explicitly, where we do cue them and see whether they will use it. But we haven't done that yet.
Thank you. I think last question from Laura. Do you know what is in nature the nectar concentration preferred for hummingbirds, and how that might affect the choices when they're faced with the different values of sugar concentration?
Yeah, so we have done that, and had a student who that was her thesis, was actually very, very interested in the concentrations. So we've discovered that birds, individuals either prefer when you give them all the possible concentrations, it's about 35. They either prefer 35 or 45%. Once it gets up to 55%, we think it's they're slower to take it up. It gets much more viscous. So there is a drop off. It doesn't keep going up. So somewhere in there is a trade-off between the time taken to take an increasingly viscous solution up. We've typically used 20-25% in our flowers, and the birds work for that. So we keep with that, because then they come back about every 10 minutes through the whole day, and we can also offer them many more flowers than if it was 45%. We know they stay away longer the richer the food is. So again, if we want to increase our number of trials, we decrease what we're giving them.
Great, thank you. So that's time for the second talk. I will close the videos of Susan and Rory, if that's okay. And Lars, please start sharing the screen.
Can you see my top slide now? Oh, all right.
Yes.
Okay, great. So what's the time now? 42. All right. So before I start talking about whether bees could be said to have a mind that is useful in foraging, I'll thank my co-authors, Catherine Wilson, Ellie Leadbeater, Quinn Solvey, Ollie Lukola, Joe Woodgate. Okay. So just to reiterate, I guess, a bit what flower visiting animals must accomplish when foraging. So in any bee's flight range, I guess just as in a hummingbird's, there are typically dozens of flower species that might all differ in reward, concentration, and quantity, in the accessibility of these rewards by way of complex or not so complex floral structures, in distances to the home base that must be taken into account, and so on. And bees have to figure out not just where these flowers are, but also to associate those most rewarding, the signals that are most, of the flowers that are most rewarding, and then predominantly focus their efforts on these while using their attentional processes to disregard other flowers. It's all not a small task if you have the brain the size of a pinhead. Now, the way people used to approach foraging questions, and I'm sure Alex Koselnik already set you right on that last week, was that in some models they either had no information, and others they had all the information, but often what you'd see is something like this. So here's a distribution of favorable areas food-wise or ion concentration-wise for an amoeba. The blue areas are the favorable ones and the red ones are the not so good ones, and that the animal or amoeba then moves by making short movements and sharp turns in areas that are favorable in all these blue areas. And when it finds itself in an area that is not so good, it just makes straight and long movements. And I have endless arguments with the modelers with whom I collaborate, who still think that you can apply these kinds of mindless foraging models where the animals know nothing to anything from bees to albatrosses, even humans, and also including bacteria. There are other models where the assumption is that they know everything. And that, of course, is also perhaps, even if it sometimes you can stick a formula on it and think that correlates with the movements, a bit simplistic. The idea that I want to run by you, and that's an emerging idea in my lab, and we're just collecting tentative evidence in that direction, is that foragers gradually acquire information and then can actually think about it. And then they can juggle multiple memories, compare options in their minds, that they have representations of spatial locations and things, that they can make predictions of the outcomes of their own actions, and also that there are at least basic forms of emotion in pollinating insects such as bees. So just to make you aware of just how tall a task the navigation in itself is, if you find yourself in a foreign city that you've never visited before and you actually turn your sat nav off for a little bit, then it often takes quite a bit, and you don't have a map, for example, then it can be quite easy to get lost. However, it's also useful to remember that orientation in cities is actually facilitated by memorable landmarks, buildings that are made to be memorable and to make navigation easier. The typical environment that a bee or a hummingbird might navigate in is perhaps more something like this. So you might find that your home base, your nest, is over on one single tree here on the right. There might be a flower patch with good pollen quality down here in that little valley, and another good flower patch a kilometer behind that hill. And there's a lot of aliasing in there. There are lots of trees that will look the same or similar along the way between these destinations. And it's not a trivial task to find your way through such environments, as any hiker who's actually tried to navigate without any kind of aids or a map will probably have discovered. So the way we study bee navigation is with this harmonic radar tool, where one dish sends out a signal that is then bounced back from a transponder that's glued to a bee's back, and then picked up by another radar dish to inform us about the bee's whereabouts. And we now have a system by which we can track bees in 3 dimensions by having a radar that can look at various heights as well, and computing a 3D signal. And what we can do with this kind of equipment is not just track a bee in 3 dimensions. This is a flight of a honeybee drone here, by the way. But we can also then take the view from the cockpit of the bee as she's navigating through her environment, which will allow us to figure out not just what the bee sees, but also what she chooses to see. What kind of landmarks are interesting for the bee? Does she choose to forage in landscapes that are more easily memorable, and so on? By the way, can you all still hear me? It's always bizarre to talk to your blank screen. Am I audible?
It's
all right.
Yes, I think,
Right.
And the videos look great.
And what we can do with this kind of setup is asking such questions as, I'm glad Healy already introduced the traveling salesman problem and trapline. So here's a simple traveling salesman task. Here's the hive, and there are 5 flowers, which in their sum have just enough reward volumes for the bee to fill up once. So she has to find an efficient route between these 5 locations, minimizing travel distance and time. And what we can do is we track a bee from its first flight ever, trying to solve this task over multiple dozens to see how she gradually improves. And if we do that, this is what you'll see. So here's the bee. She's found the first 2 flowers at the stage. The bout number is shown at the bottom, at the top left here. So now we're in bout number 3. She's discovered 2 more flowers and the 5th one. But you can see the tracks are still all over the place. But gradually there's a streamlining of solutions. She's still exploring outside locations, but she's getting better and better until over time she actually finds the optimal solution to this route. So using this technology, we can not just look at which sequences the bees visit the flowers in, but also how they get there, what they do between the feeding stations. And you can see that this is very different from any kind of near-far search algorithm. Everything in the final track here is, of course, determined by memory. The bee now knows the positions and links them with flight segments that minimize overall travel distance. Now, what we can also do is leave the bees to their own devices. In this case, we have no artificial feeders, and we just track bees' entire biographies, their entire flight behavior from their maiden flight through their entire lifetime. This is what we did here. This is that bee's very first flight ever. The overall flight takes over 2 hours. The blue dot here is the hive location. The track fades to, or changes to red as it's getting older. It's further in history. This is all still the same first flight. The bee loops in different directions, exploring various locations, and always centering back on the hive without entering it, then trying typically another direction. Now she's up at a forest edge here, which will be interesting a little bit later. Just memorize for now that she knows this location because she's been there once. Dashed lines are when we temporarily lost the bee off the radar. Now she's back in the nest. The whole flight took over 2 hours. Now what happens on the next day? This is all the bouts number 3 to 82. So we're now following it for several days, just that one individual. Green is early, red is late. And so the bee does one more loop in this southwesterly direction, and then she's discovered something. And now for several days, she continues visiting just one flower patch and just like an assembly line worker does nothing but back and forth for several days. Now, after a few days of inclement weather, when the bee stayed inside, she does a few more bouts out to this location here that she's visited over the first week of her career. And then suddenly midway appears to change her mind and flies to a destination that she'd only explored a single time on her exploration flight 10 days earlier. Now, this is just anecdotal data in that it's observational and it's only a single bee in this particular case, but there seems to be a retrieval of a distant memory that she retrieved without flying back to the hive, but changed direction halfway through and then looked at or dedicated her foraging efforts to another patch that perhaps was now superior to the one that she previously visited, until then for several days just continues with this particular forest patch until on day 13 she just vanished off the radar, presumably having been eaten by a bird or a crab spider, for example. Now, one thing that foragers can also do, in addition to exploring for themselves, is to look at what others are doing. So if you're an entirely naive bee, then a useful starting point might be to look at which flower patches or flower species there's particularly high activity by other bees, because that indicates that these have already invested into collecting information about a suitable patch and have found it. So what you can do is take this sort of scenario into the laboratory, and this is what Healy Leadbeater and others have done, where in this case, in this flight arena here, you can see there are plenty of bees visiting orange, but not green, and there's a bee behind a screen here observing, but she's not allowed to fly in. And you can do also the reverse, where the green flowers are occupied by foragers and the orange ones are not. And then you clean everything, you remove the experienced foragers, and you face that observing bee with a choice. Will she now pick the orange ones or the green ones, depending on where she's seen foraging activity? It turns out that indeed these observing foragers will subsequently visit these options that they have seen from a distance without receiving reward at the same time, other bees foraging on. Now, this could be explained by a relatively simple form of, a two-step form of associative learning. So it might be simple in that what the bee might be doing is two association steps. So the bee might first associate conspecifics with nectar if she's previously foraged jointly with them. And so in that case, the appearance of a conspecific comes to be associated with nectar as the unconditioned stimulus. Then that bee is in that observation chamber. She sees conspecifics. Conspecifics mean reward. She sees them with a particular flower type. And then she connects the two in her mind without at that time receiving a reward. And the flower, which is at this stage still not associated with something rewarding, but with another stimulus that's associated with reward. And then that means for the bee, this flower is nectar rich, because I know that there have been bees on it, and bees mean that there is something to forage from. So this particular form of foraging, where you've seen other foragers, might be explained by associative learning. It might be simple in that sense, but albeit in a kind of two-step manner. But there are other forms of social learning where it's perhaps not so simple. So here's a task of object manipulation, where the bee has to roll this little ball into the center of a horizontal arena. And they can learn that, all right, so she picks it, rolls it backwards and moves it to the goal area, a reward. So far, so good. But what about social learning? So we had a situation as follows. So there are now two bees. One is the experienced bee, this one, and this is a naive observer that's never rolled balls for reward or otherwise. It just is let in there with a skilled demonstrator to observe what's being done. And you can see there are 3 balls here. Now, obviously, the optimal way to solve the task is to pick the closest ball and move that to the center. But that's not what the demonstrator does, because we've trained that demonstrator to know that this ball and this ball, the 2 closest ones, are glued down. They can't be moved. So what the demonstrator does is always move the furthest ball, and that is also what the observer then gets to see. And so this is one of the 3 trials. So here's the experienced individual rolling the ball. And then at the end of that, once it's in the target area, both of them get a reward. The question then is, so what the observer has now seen is a solution to the task that is actually suboptimal. It's rolling the furthest ball to the center. And now what happens if you then put the observer on the spot on its own and ask it, well, what do you do? Which ball do you roll? Do you ape the demonstrator? Do you copy what you've seen it do and pick the furthest ball? Or do you pick the closest ball, which would actually be the better solution? And she picks the closest ball. She's a bit clumsy, as you can see, but clearly she's this and all other bees picked either the closest or the second closest ball, not the one that was the furthest away. And that indicates to us that it's a little more than associative learning. It indicates to us that there is a kind of a form of understanding of what the desired outcome of the action is, instead of just copying stupidly the actions of the demonstrator bee. Now, we know that bees are good pattern recognizers. Obviously, they find the flowers that they have been conditioned to by the rewards that they find in there. But it's actually very difficult to show that they're learning these images as patterns, that they're storing little images in their brains, because many of these pattern recognition tasks could actually be accomplished with very simple feature detectors that just measure the orientation of edges of certain colors and so on. So one way to explore whether there's actually some sort of internal representation of an object shape is by testing cross-modal recognition. What we did in this experiment is we trained bees to distinguish 2 types of shapes, balls and cubes. But in this case, the bees, when they were trained to recognize them under lit conditions, they could not touch them. So they had to land on top of a plexiglass lid and then would find, in this case, for example, that the balls would contain a reward, the cubes would not. And we would then test the same bees in darkness, shuffling the positions, of course, of the targets, and see if they could now find the same objects without seeing them, but by touching them. So in that dark scenario, they could actually hug essentially the shape, they could feel it, whereas they could not in the lighted scenario. And so the bees trained on balls in the light managed to find balls in the dark by touch. And we did the reverse experiment as well, where we trained the bees in the dark and then tested them under lit conditions in the light, and again they managed to find the correct shape. And we, of course, we had multiple bees per treatment, and a bunch of bees were also trained on the reverse task, where they were rewarded on cubes and found no rewards on the ball. So that indicates to us that there is some form. There's not just a recognition by simple feature detectors, but one that actually resorts to some common representation of object shape that's accessible to multiple sensory modalities. Finally, there is an experiment that I wanted to run by you, which seems to us to indicate that there is at least some form of basic emotion-like state in the bees, and it's essentially a task asking whether a glass is half full or half empty. So this is physically an ambiguous stimulus, and depending on whether you're pessimistic or optimistic, you might judge that either way. I think Rui Oliveira, who seems to be in the audience, did that recently with fish. Here we did it with bees. So what we trained the bees to learn was that, for example, in this little flight arena here, the left blue option is associated with reward, and the right green option is not. And so they were trained for that a number of times. And this is a go, no-go task. There's only ever one option present, and the bee can decide whether or not it chooses that single option. And so in training, this is what the bees were presented with. During tests, we gave the bees various ambiguous options. So this is an intermediate glass half empty or full task where there's no reward present now in the testing with these intermediate options, and the bee has to judge whether this ambiguous stimulus is more likely rewarding or is it perhaps more likely unrewarding. And this is what you observe after you've trained the bees. It's that you observe that bees will typically fly very swiftly to the option that they've come to associate to learn as rewarding without any hesitation. Whereas if you give them green and nothing else, then they will faff about for some time. They'll eventually accept it. Maybe there's something there after all, but it will take them quite some time to accept that option. Now, what do they do with intermediate options? So this is an ambiguous option. It's turquoise, halfway between blue and green, and we then again measure the time that bees spend before they're accepting this particular option. And we did a little manipulation there in that when we gave some bees a little surprise reward before they even entered the flight arena. So they were given 5 microliters of sucrose solution before they entered the setup. So this was a kind of surprise reward. And we basically then asked, would these bees that have now been given a little extra, a reward in a location where they didn't expect it and had never experienced it before, whether these bees would behave differently to bees that were just stopped in the same location but received no reward. And this is what we found. So the position P is the rewarding option, and invariably bees accepted with a very small latency of about 10 seconds. Whereas the non-rewarding option at the other end of the scale, so this is when we show them green, no matter if they've had a reward or not before, they will take almost 2 minutes to explore that option. So clearly they've learned that this is very unlikely to be rewarded. What's interesting is what happens at the intermediate options. And for those bees that have received that little sucrose droplet as a surprise, invariably for the intermediate options, whether they're turquoise or a little more blue or a little more green, the latencies are shorter. That's the red line here. So these bees accept the intermediate options more as if the glass was half full, whereas the ones that have not received the surprise reward, that's the black line here, would have longer latencies and take longer to accept these intermediate options. So for them, the glass was clearly half empty. This, by the way, is the only data figure in my talk, so I hope that wasn't too taxing. There are also experiments by Jerry Wright on and Melissa Bateson on the converse scenario where bees had a surprise simulated predator attack, and in that case, you will find the reverse. You will find them to be more hesitant to accept intermediate options. All right, so a few conclusions about these explorations of what we think are some novel dimensions of the world of foraging. So we think that bees have representations of space and things in space, as well as time and number, which I've not talked about here, that they can learn by observation in a manner that is not just associative learning, because there's some evidence, at least, that there is an understanding of the outcome of the the task. We think that bees have flexible access to their memory libraries, basic emotional states, and an idea of what they want, and using a kind of awareness of the possible outcomes of their own actions. Now, none of this, I guess, is formal evidence for thinking or consciousness or even sentience, but I think as we accumulate more and more pieces of the puzzle, we're nudging the probabilities in that direction. You could implement all of these abilities in a machine learning algorithm or a robotic solution, but you'd have to program your machine in such a way to actually solve these tasks. They wouldn't come by these solutions by themselves. So there's, I think, a crucial difference there. All right, if that's at all of interest, there's a popular scientific essay that I've written with philosopher Catherine Wilson on this topic, and I'll stop waffling now. I'm looking forward to your questions.
Thank you very much. That was really fascinating. I think we have one like kind of technical question from Sergey. Sorry, a very naive question. How do you track the bees in natural settings? And a follow-up. Yeah, sorry, go ahead.
So the equipment is called harmonic radar. The bees are too small to glue a transmitter on them. So anything battery powered won't work. What we do instead is we use what is called a transponder, as I showed in this one image where it looked like there was a little antenna on the bee's back. And that's a transponder called so because it actually picks up a frequency from one of our radar dishes and transponds it to a harmonic, twice the frequency actually, which is then collected by the second radar dish. So that's how we do that. But that also means that we can only ever track one bee at a time. So it's
Great. That was the follow-up question, whether it would be one at a time. A second question, do you think the emotional bias in the response latency of the free-flying bee is dose-dependent on the sucrose concentration and quantity provided? This is a bee asking.
It's possible. It's possible that you'd get even more excitement if we had given them more. We tried to keep it small because, on the one hand, we wanted to keep the bees motivated to forage for more. It was just meant to be a little surprising event for them as they walked towards the arena.
All right. I think these are currently the questions we have for the talk specifically. I'm starting to invite the other panelists and Susan. Sorry, is there? There's another one from Charlotte. Both fascinating talks. Genuinely curious, how long does it take to train the bees to move those balls? Do they learn to do this? Do they all learn to do this? This is again for you.
Yeah, it depends on the training method. The best method to learn is by them observing other bees. And so the experiment that I just showed you was one of 3 learning trials for that observer bee to know what to do, although such a bee is still very clumsy at actually doing it by themselves, especially if they have to roll it over a longer distance. But 3 observations are enough to learn the bee in the ball in a skillful kind of manner. Takes a few dozen trials, but the understanding where the ball goes is very quick. Do all bees learn it? So for whatever task we use, there is huge variation between individuals. And so we had, I think, in the introduction, you mentioned the string pulling task. So that was a task where we trained over 100 bees, and the vast majority of them learned it either by observing skilled individuals or by us training the bees in a stepwise manner. There were, however, 2 individuals, only 2, that learned the task without any training, that solved it spontaneously.
It's really fascinating, I think. So everyone is back here. So the idea was to open up the discussion kind of a little more broadly, sort of playing off themes that came up in both talks. So again, we're inviting questions from the audience, and otherwise I think we have a number of questions as well. Should we start with one of our questions? Anyone wants to ask a question?
I mean, can I ask something that's related to what Lars was just speaking about, but also that I think that relates to both this idea of like individual differences, actually, I think is something that's very interesting. And I actually was wondering whether the hummingbirds, for example, exhibit individual differences as well, or whether they also, for example, just have different learning rates, that kind of thing. Like, what else might actually shape learning rates in both bees and hummingbirds? I think is just interesting.
Do you want to start, Sue?
Sure. Just sure. So we haven't explicitly looked at that. We look at what the average boy does, as it were. But we do know that some males take longer to learn. The problem is, particularly in the field, but it's true across the way, is that the time it takes to learn something is so totally bound up with motivation that I wouldn't want to say anything at all about, above and beyond that. So that's the first thing. And what was the other thing that I was going to say about that? No, that's the first. Yeah, I'll come back to what I was going to say as well. So Lars, do you want to? What's the other thing I was going to say?
Multiple ways of looking at that. So there is an evolutionary perspective here. So we did one study where we tested individuals from multiple colonies in a simple flower color learning task. And there were not just individual differences within a colony, but there were also recognizable colony signatures. So that essentially some bee families, and of course in a beehive, they're all very closely related. All the individuals tend to be very slow, and there are other colonies where all the individuals tend to be very fast. And we then took the same colonies, the same genetic makeup, and put them from the laboratory in the wild and measured their foraging efficiency by weighing the bees as they flew out and then returned to the hive. And it turned out that the colonies with the faster learners were also on average those that brought back more rewards. Another question is more mechanistic. What on a neural level, what kind of differences do you find? And that was a study we did a few years ago with a PhD student, Healy, who found that the microglomerular complexes in the mushroom bodies at the intersection between visual projection neurons and the mushroom body's Kenyan cells, in those bees that learned faster were denser to start with, so there was a higher density of these synaptic complexes, and also they increased faster as a result of further learning. So there was a mechanistic correlate of high learning speed there.
Okay, so I have sorry, can I just a couple
more follow up
hummingbirds. The first of all, that we haven't yet had a male that hasn't learnt to do our experiments. So we now have nearly 30 years of doing experiments and haven't yet had a male that can't do what we do, what we have asked them, except on instances where everybody doesn't do it or can't do it. And then we realize it's something about what we're asking of them, not what they can do. Very clear differences between territorial males and males that don't have territories. And so the cues they use, for example, the cues they prefer to use, our floating males use visual cues over spatial cues, first of all, and they're the ones who find the feeder hanging on your chair, whereas the territorial male goes back to where the feeder was, even when it's not there. Things like that, which are really clear differences. Very, very different foraging too, actually, in the sense that our territorial males only fill, we haven't collected these data, but other people have, fill their crop to about a third. So they are not on the edge, by the way. They're absolutely not on the edge. Non-territorial males are, they drink and 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 look like they're, you know, they get the opportunity, they fill up. So do females, that females do the same thing. And the final thing I want to say is that when we look at navigation and the cues that, or, you know, how animals are, what information they're using to get back to flowers, then we see differences across males that are much more striking, that individuals vary in how good they are at relocating very accurately in space and what information they use to do that.
So I think this is kind of a related question to this, what you've just described. And I don't know, to me, there seemed to be, again, kind of a similar theme in the two talks with the hummingbirds. One of the early experiments in your talk that you described where it was sort of the conflict between local and what you're calling spatial cues, where I was also curious what you know about, you know, the nature of the spatial cues. And then actually maybe most prominently this experiment with the bees learning shape in the, like, visually and in darkness. I mean, it seems like they are learning, right? Like I was curious, like they learn these different the information from different sensory cues, and it's unclear to me how much these different bits of information are combined into one thing, right? Like one representation of where the food is, say, or which flower is good, or versus then how much you can use them separately if they start being in conflict, or I don't know, it's more useful in a different task.
So maybe I'll go first, because I just want to say I'm totally envious of most of Lars's experiments, but not least because of the being able to test his animals in the dark and in the light. Our birds go to sleep in the dark, so come nighttime they go to bed, and that's that. So it's a bit disappointing, but otherwise I obviously just try to copy Lars's experiments as frequently as possible. But what our hummingbirds will do is combine different visual and different, what does different mean? But different kinds of information. So I said before, they can remember where a flower is, they can remember what the color was, and when it is to be rewarded. So you can put out flowers in other places of the same color, and we did in an experiment where we in the morning had an array out, and the birds had to go, for example, to the yellow flower in the bottom hand corner. In the afternoon, the arrays are out, and the bird has to go to the blue flower in the upper corner, and they do that very, very quickly. They can put all three things together, and then you can look at their mistakes. So actually, I've got very interested in looking at mistakes, not just when animals get things right, because we learn a lot, maybe even more from the mistakes they make than we do, because when they get something right, it's a bit like a student gets the answer right. You have no idea how they got there, but when they make mistakes, you can start to see, which is why in maths, my guess is I want to see the working, because you could see the logic of what they're doing. And again, with our animals, when you see, so for example, when we've got these experiments where they can put all three things together, the most commonly made mistake is to the wrong time, and they find that really hard to correct. But if they go to the flower, the one that they find really easy to correct is when they've made the wrong color decision, and then it's like, oh, the next visit is always the right one. So there's certain mistakes they sort out very quickly, and other ones they just don't know that. Yeah.
Yeah, so just to expand on the second half of your question. So the traditional way, I guess, of at least how bee people have asked such question is that they would, let's say, present a compound stimulus of a color and a shape, and then to see how bees would respond if you give them just one of these cues, or if you give them in combination or put them in conflict. And sure enough, they will recognize what they've been trained to, mostly in combination. That seems deceptively clear, but it's actually possible to mimic such performances with very simple feature detectors, still without there being any kind of internal representation of the bound features together. Very, very simple computational mechanisms could do that. What we did in this particular experiment with the cubes and balls, where the bees were not taught in any way to associate the cubes in both scenarios with reward. Okay, so they were not taught the ball shape is rewarding in the dark and it's also rewarding as a visible target in the light. They were only taught in one sensory modality. So the bees that were faced with balls to touch and cubes to touch had never touched them before. They had only seen them through a screen, right? So it had to be a spontaneous generalization of that feature from one sensory modality to another. They were not trained to associate both sides of the mechanosensory and the visual during training. When they were presented with the task, feeling the stimuli in darkness when they'd previously only seen them, they had never felt them before, and vice versa.
That's nice.
That, I think, is what points to a kind of, at least that's the explanation that we see there, is that there must be some sort of representation of shape features.
Brilliant, thanks. We also have a question in the question box from our first speaker, Alex Koselnik, which I thought I could ask you. So he says, people in AI use both model-free and model-based learning in decision processes. Many of your results imply that model-free algorithms will fail, or be difficult to design to solve the same tasks as the animals. What does the addition of the term consciousness add to the difference between algorithms with or without representations of the relevant affordances in the surrounding world? Is there any novel prediction derived from attributing consciousness? It's a brilliant and quite long question, so you can take some.
I'm guessing that's a question for me. So I'm not sure. You've illuminated my ignorance with respect to these algorithms, but I think the tasks that I predict you will have difficulty implementing in any machine, no matter what kind of computational project it is the more flexible problem-solving tasks. So you can program machines to solve all kinds of things, and I'm sure you could easily program a machine to do everything that we knew bees could do, or hummingbirds could do, or crows could do 10 years ago perfectly. I don't think that there's anything that was known 10 years ago that you couldn't model. But most likely, as far as I can see, that machine would have failed on everything that we've discovered hummingbirds and bees and crows to do in the last 10 years. If you build such a machine today, you could build a machine that did everything I just told you about bees, but it'll fail on what we'll discover next year. That's my prediction anyway. So I think what consciousness allows is a kind of more flexible exploration of possible scenarios, and therefore more flexible problem-solving solutions than we currently can muster with machines.
Okay, so I have the honor to maybe ask the last question of the seminar. So just to arcing back to the title Foraging at the Limit, and maybe also connecting it to the next, I guess, 2 episodes that we have, I was wondering what are really the limits of foraging in terms of the cognitive capabilities, and are there like any possible like heuristics that the animals can use to solve these kind of problems? Have you seen any evidence for that in your data? And maybe also if the animals are challenged by, for example, being starved or deprived from certain things, do the animals, for example, make more mistakes, as Susan was explaining for certain individuals? Like, did you test that in your experiments in any way?
So I mean, unfortunately, we can't do that very easily in the field. It's very difficult to manipulate the state of the animal. We can manipulate what we give them, and they respond differently. But it's really hard to get to that. In terms of the limit, I think this is a really interesting question, and I don't have an answer. But I have some stuff with which you can do as you like. But the first is about brains, and so Lars has made a comment about the size of a bee brain being the size of a pinhead, and Andy, Healy and I have talked about our hummingbirds having a brain the size somewhere between the size of a grain of rice and a baked bean. So relatively small. Well, not relatively small, actually. Hummingbirds, for their body size, have a relatively big brain, but they're small. They're just absolutely small, sorry, not relatively small. And yet, when we've asked them questions that we try to have them based in something that comes from what we perceive to be hummingbird foraging-like questions. So we're not asking them, what was the latest book they read, but they've answered it, and they learn the question we're asking them, and they give us an answer that seems to answer us reasonably appropriately. And so the more I work with them, the more I wonder what the heck we're doing with this. We have a very large brain, and it's not obvious to me that we are putting it to as much effort as the hummingbirds are managing to get out of their much smaller brain. But we then have to think hard about natural selection. Those animals, their mistakes are very costly, and it's any single foraging trip may or may not be costly, because they are making many foraging trips in a day. So any single one is not that costly. But we saw with our hummingbirds that even, as I said, even when the scale of the experiment is really small, they minimize the costs of that. Because if they get it wrong, then they're toast. And whether they are, they're very likely to be much closer to natural selection and its costs than certainly I am. I go to the fridge for my food. I don't have to find food in quite the same way. There are humans who don't have a fridge for the food, but again, I think I'm just throwing things up in the air. It's on the edge, as I said. Our territorial birds are putting their feet up. I mean, they are not right on the edge, but they're managing their lives, so they don't have to be on the edge, at least for the time that they're at our field site. It's hard to do the kinds of experiments to really nail some of the things that you would like to. That is for the lab, where you can, and some of Alex's work has shown really beautifully that, what animals do differently depending on the state they're in. And they definitely use heuristics. They will be doing cheaper ways of getting to stuff.
I think in terms of what are the limits of what you can do with very small brains, I think people commonly underestimate both the storage capacity as well as the flexibility that you could achieve with very small nervous systems. So Barbara Webb, for example, has built some brilliant models of ant foraging. And so she works with ants that navigate through very cluttered sort of bush environments and so on. And she built some actually quite reduced models of the complexity that we already know exists in ant brains, and found that these ants can easily store and distinguish 80 different snapshots of the environment on a single foraging path, and discriminate these scenes. They're highly cluttered environments, very similar scenes, and a very reduced model. 80 images, no problem. So if you're asking about limits of storage capacity in nervous systems, I think there's a lot to be learned from these small-brained animals.
I would like to ask a maybe brief question that's a little bit related to the earlier question on, consciousness and model-free, model-based learning. I think the thing that came up were like emotions, and I was curious if is this? I mean, what is the? I think for kind of efficient and flexible, quickly adaptive learning of strategies, right? It seems essential that you have some way of telling whether what you are doing is working, and I mean, sort of some evaluation of what you expect versus what's happening. Is that where, I mean, that's kind of how I interpreted that sort of role of emotion. I mean, maybe you can elaborate a little bit, and whether you saw similar things in hummingbirds, or some sense that they had an expectation and you could see this maybe in the behavior even.
Oh yes, there are many. So yeah, if we switch out sucrose in the flower that they've been to once, they only have to do one. They do one trial learning, and they learn a location. And switch it with water, then their response is it's pretty violent. Because they put their tongue further in. Once they know it's there, they put their tongue further in, and then they get a mouthful of water, and it will just come spitting back out. And sometimes they come over and display to us. Okay, they have that experience and then come straight to where we are, and then will display at us. And there are times, if you want to get very anthropomorphic, where it would be like, they're coming and throwing the fist at us. But yeah,
that's really fascinating.
Absolutely.
So was that a fair? Sorry, just to ask this: was this a fair interpretation? What do you think, Lars, would be a use for having these emotions to help switch strategy? Is that reasonable?
I mean, first of all, of course, we have to be entirely clear that I and others are on very speculative ground. So there is no formal way to diagnose emotions in any animal or consciousness, for that matter. All we can say is that by the same criteria that are used for asking whether a domestic animal, a goat or a cow, in a given farmer setting is optimistic or pessimistic, and whether it's a happy animal or not. We're basically using the same paradigm. The animals can't tell us, only indirectly through these kinds of tests. So to the extent that we believe these criteria in a goat, and using the same criteria, copy and paste on an insect, if it qualifies by the same criteria, then we either have to accept that there is something equivalent to the goat stage in a bee, or we have to dismiss the criterion for both animals. But it seems to me that a lot of what we observe in a way as Sue has just described does not make much sense without any kind of emotional states in these animals. And again, you could build an argument that the responses that you see, well, let's take a scary scenario when You use fear in conditioning experiments, for example, in rodents and so on, and you see all the kinds of responses that you would also see in a human. Now, you could, with the exception of the verbal report, you could build a scenario, well, this could still just all be reflexes. Maybe so, but I don't think nature has room for such profligacy. You wouldn't build these kinds of overt behavioral responses if they weren't a reflection of some internal state just to fool us humans. Now, in our bees, we don't have the same kind of responses as you might find in a mammal of squealing and grimacing and so on. But the scenario that people up until a few decades ago is that there is just not even basic nociception in these animals is almost certainly wrong. Whether there's a subjective experience of fear or suffering, we don't know. But it's certainly plausible, and I think it does make some biological sense to have an unpleasant response in the face of an essential threat, for example, is not something for which you need to necessarily be clever. It just makes sense to increase the motivation for you to get out of the range of the threat. But I'm speculating here.
Yeah, certainly if you look in a bird brain, you see all the areas that you'd expect to see, or that you do see in a mammal, in terms of the amygdala and various brain parts that are associated with what we would call emotions, and many of them, as Lars has just said, would be useful attributes for an animal to have. But then I was thinking, what about being happy? So you go through the list, right? And what about being happy? Well, they certainly have reward centers. So whether or not they would feel happy as the way we manifest it is not the same. But may not be the same. But there are definitely reward centers that are turning on when some behaviors are happening. So yeah. In my nest building birds, the reward center has lower activity when male zebra finches are tucking in material into their nest. And so we take that as meaning that the males don't like doing this, that this is the sort of tidying up role that they'd rather not be doing. But again, slightly flippant. It's a totally serious question.
No, there's so much to explore. I think this was really fascinating. I love, seeing these two perspectives, remarkable similarities, I would say. Sorry for all the questions we did not answer. But yeah, I want to thank again both speakers. I think it's time to wrap up for us.
All right, thank you guys very much.
Thank you very much for your
Thank you.
All right.
Thanks everyone. Great talks, and thanks for everyone watching as well.
Thanks.
Okay. Have a good day.
Nice day or evening or morning, wherever you are.
Yeah.
Yes.
All right.
See you in a couple of weeks, everyone.
Okay.